Hello. Very good afternoon to the student who have joined. Can you let me know in the chat box if my voice is audible to you? And also, please let me know if you can see the presentation which I am sharing in the chat box. Kindly let me know if the voice and audio. Okay. Thank you. And you can see the presentation also, right? Let me know in the chat box. So, very good afternoon to the students. Please wait for the some time. At 4 p.m. we will start our session. Meanwhile, please let me know if you can see the presentation and also if you can hear to my voice clearly. We will wait till it's 4 p.m. and then we will start today's session. So please wait till then. Okay, so it's almost 4 p.m. I will wait for 1 more minute and then I will start the session. Okay. Meanwhile, kindly let me know if there is any problem with the audio and also with the video. And let me know if you can see the presentation clearly when I am sharing it. Okay, wait, just I will make the call open. Yeah. Okay, so it's 4 p.m. Let me start with the session. Meanwhile, when other students will join, they can directly join into this class. So welcome to the week 3 live session of the NPTEL course of Medicinal Chemistry. My name is Mousumi Deb and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this course of Medicinal Chemistry. And today it is our third live session. So before starting the session, every week I'm just to summarize a few points. So let me start with that. So there will be this kind of problem solving session every week from on every Friday from 4 to 6 p.m. We will continue this type of live session up to the 12 week because this is a 12-week course. So for each week course content, I will take a live session of 2 hours from Friday 4 to 6. And also at the last of the session, we will have a summary session when I will summarize the complete course and we will solve some problems so that it will be helpful for you also. So all these sessions are live on Google Meet platform. The link is same, which the link you have used today for joining. This link is same for every class, every Friday class. So you can save it anywhere, and then you can use this to join the class. So the main point is, if there is any network disruption happen from my side, please wait for some time. Do not leave the class session immediately. I will rejoin as soon as possible, and I am recording this session so that because I want to record it seamlessly, that is why I have muted your microphones and also the cameras, so that there is no issue in recording, because this recording I have to share and with you after the class. But the chat box is always open. I am monitoring the chat box every now and then. So even every point, every doubt, every thing, if you want to tell me anything, you can post it your doubts and queries in the chat box. Okay, I will check it in time to time, and if still after the class the doubt or the problem persists, you can drop an email to me. I will share my email ID also at the end of the session. And since I'm recording the session, if there is any network issue from your side, you can leave and you can rejoin at any time, because I am recording the session and I will share the recording after the class. So you can check it from there, the part you have missed. And as I have already mentioned it many times, that this presentation which I am presenting today and the recording, it will be shared with you after the class. Okay, and I think I have mentioned already where to find these recordings. Okay, so you can find the recording. I will just show you at the end of the session. So what we will do in this live session, so in this live sessions, we will have around 15 to 20 questions regarding the course content, which is live on that time. So today it is the third live session where we will discuss the course content, which is in the third week of or the week 3 of this course of medicinal chemistry. Okay, the topics which are in the week 3 of medicinal chemistry, we will discuss those topics, and I will share with you around 20 questions. All these questions will be MCQs, just like you have in your assignments, your weekly assignments. All these questions will have 4 options, A, B, C, or D, and you have to figure out which of the following options will be correct, and you have to let me know in the chat box. After showing one question, I will wait for some time and I will check the chat box to see which answer you have given, and then I will show you the answer. After showing you the answer, I will discuss that particular topic in detail, so that now, in the final examination where you have to face subjective questions, you can answer those questions easily. Okay, so just let me check if how many students have joined. It's already 4 p.m. Okay, let me start with the sessions. I think most students will join eventually. So the topics today which we will discussing today's are, this is in the week 3 of this course of medicinal chemistry, is the nucleic acids, then RNA and protein synthesis, then nucleic acid basics or molecular biology, enzymes as drug targets, enzyme kinetics and inhibition, and enzyme kinetics and various mode of inhibition. So today mainly we will discuss regarding the nucleic acid, RNA, protein synthesis, some basic questions regarding the molecular biology, and also we will discuss regarding your enzyme inhibition, types of enzyme inhibition, and their examples. All these things which we will discussing today. Okay, so let me start with the session. Let me show you the first question of today's session. So the first question of today's session is, which of the following statements is correct? Is incorrect, sorry. Which of the following statements is incorrect? Your options are: number A, the distance between two major groups of DNA is 34 angstrom. Number B, the distance between two major groups of DNA is 10 angstrom. Number C, uracil is not present in DNA but is present in RNA. And number D, linkage of nucleosides happens through the phosphate group. Again, I will be repeating. Which of the following statements is incorrect? Your options are: number A, the distance between two major groups of DNA is 34 angstrom. Number B, the distance between two major groups of DNA is 10 angstrom. Number C, uracil is not present in DNA but is present in RNA. And number D, linkage of nucleosides happens through the phosphate group. So you have to let me know A, B, C, or D, which of the following statements is incorrect. I will waiting for some time, and then I will check the chat box. After checking your answers, I will show you what is the correct answer, and we will discuss this question in detail. Which of the following statements is incorrect? Number A, distance between two major groups of DNA is 34 angstrom. Number B, distance between two major groups of DNA is 10 angstrom. Number C, uracil is not present in DNA but is present in RNA. Number D, linkage of nucleosides happens through the phosphate group. A, B, C or D, which of the following statements is incorrect? You have to let me know in the chat box. In the class with the questions which I am showing, there will not be any marking. Okay, so even if your answer is wrong, it does not matter, but answering in the during the class is very important. The chat box is open. You have to just write A, B, C or D, which of the following statements you think is incorrect regarding this particular question. And when I will check the answer, even if your answer is wrong, it's fine, but answering is very important, otherwise I won't understand that you are following the class or not. These types of MCQs are in your assignments. So answering in the class will also help you to solve the assignments part. I will wait for 1 more minute and then I will check the chat box and show you the answer. So which of the following statements is incorrect? Let me check the chat box. Okay, I have one answer which says it's number D. Others? Anyone else who wants to answer? Again, I will repeat the question. Which of the following statements is incorrect? You have to find the statement which is false. Your options are number A, the distance between two major groups of DNA is 34 angstrom. Number B, the distance between two major groups of the DNA is 10 angstrom. Number C, uracil is not present in DNA but is present in RNA. Number D, linkage between of nucleosides happens through the phosphate group. Which of the following statements is incorrect? Others, I only have received one answer in the chat box. I want all of you to answer the question. Let me wait for 1 more minute. Okay, let me check the chat box a final time and then I will show you the answer. Okay, so I think I have received another answer which says it's number D. Okay, now let me check if number D is the correct answer for this question. So the correct answer for this question is number B. The distance between two major groups of DNA is 10 angstrom. This statement is incorrect. But then let us start with from the beginning. So basically DNA and RNA, the building blocks for DNA and RNA, these are the known as the nucleotides. So nucleosides, okay. So what is a nucleoside? Nucleoside is basically the base and the pentose sugar. Okay, the pentose sugar which is present in RNA, it is a ribose sugar, and in the DNA it is the deoxyribose sugar. Then we have the phosphate group, which makes it the nucleoside one phosphate group makes it nucleoside monophosphate. The two phosphate group makes it nucleoside diphosphate, and the three phosphate group makes it this nucleoside triphosphate, and it is also known as the nucleotide. Now, what is the base? The base, there are 5 total bases present in the DNA and RNA. Okay, so what are the bases? So the bases are of two types. One is the pyrimidine basis. So what is the pyrimidine basis? Pyrimidine is the one 6-member ring, okay, which has the nitrogen in 1 and 3 position. Okay, and this is the cytosine, this is the thymine, and this is the uracil. So three pyrimidine bases are present in the DNA and RNA. In the DNA, there is a presence of cytosine and thymine, and in the RNA there is present in the cytosine and uracil. Okay, thymine is not present in RNA and uracil is not present in DNA. So, there is a two other bases which are known as the purine bases. So, what is a purine? Purine is a bicyclic ring. One 5-member is fused with one 6-member ring. The 5-member ring has the nitrogen in 1 and 3 position. Okay, it is like a imidazole ring, and it is basically imidazole ring and a pyrimidine ring which are fused together. Okay, and there is so this is called the adenine and this is called the guanine. So, adenine and guanine are all the adenine, guanine, cytosine, and thymine are present in DNA, and adenine, guanine, cytosine, and uracil are present in the RNA. Okay, so this is the structures of the purine and pyrimidine bases. And adenine, it binds with the thymine, and guanine binds with the cytosine in case of DNA, and adenine thymine base pairing happens in case of RNA also, but in RNA there is what is it called, thymine, sorry, not thymine is present in RNA. Adenine and uracil binding happens in RNA, and guanine and cytosine binding happens in the RNA also. Okay, so these are the DNA and RNA bases, and this is the glycosidic bond between the base and the pentose sugar, and after this this is called a glycosidic bond. Okay, and now the linkage of the nucleosides happen to the phosphate group. So one nucleoside, this is the base and this is the pentose sugar, and another base and pentose sugar, this nucleoside units both are connected with a phosphate linkage. So that is how this particular base and nucleosides are connected together through the phosphate linkage. Okay. Now this is the double helix structure of the DNA. This particular distance between this space, it is known as the minor group, and the distance between this and this is known as the major group. Now how we will understand what is the major group and what is the minor group? So let me just use the highlights. So this is one form, right? This red one is the one particular strand of the DNA, as you can see. Okay, and there is another strand. Let me just mark it in another color. Let me mark it blue. Okay, let me mark it green, so it will be easier for me. Yeah, so there is one strand and there is another strand, which is this green chain, right? So the distance between this red chain and this green chain is the 10 angstrom. It is known as the minor group, and the distance between this one green strand and green strand, or one red and red, is known as the major group, and the distance between these two major groups is the 34 angstrom. Okay, so the statement which has said that the distance between two major groups of the DNA is the 10 angstrom, this is the false. The distance between two minor groups is 10 angstrom, and the distance between two major groups is the 34 angstrom. So Watson and Crick solved the secondary structure of DNA by building a model that fitted all well-known experimental results. The structure consists of two DNA chains arranged together in a double helix of the constant diameter. This figure, the double helix has a major group and minor group, which has some importance to the action of several anti-cancer agents acting as an intercalator. The structure relies crucially on the pairing up of nucleic acid bases between two chains. Adenine pairs with thymine via 2 hydrogen bonds, whereas guanine pairs with cytosine via 3 hydrogen bonds. Thus, a bicyclic purine base is always linked with a smaller monocyclic pyrimidine base to allow the constant diameter of the double helix. The double helix is further stabilized by the fact that base pairs are stacked one upon each other, allowing the hydrophobic interaction between the faces of the heterocycle rings. The polar sugar phosphate backbone is placed outside the structure that can form favorable polar interaction with water. Okay, so the base pairs, they are facing inside of the helix, and the phosphate and this particular sugar moiety, they face outside of the helix, so that they can interact with the polar surroundings with water. And these base pairs also have not only have the hydrogen bonding interaction, but also pi stacking interaction with the above and the below base pairs. Okay, so this is all about this DNA double helix structures and the nucleosides and nucleotides. Is there any doubt with this particular first question? So the chat box is always open. So if you have any doubt, any query, you can let me know in the through the chat box. Okay. Now let me move on to the next question. So the problem number 2 is a very simple mathematical question. So it says that in a particular human DNA, it has been observed that adenine is present 27%. What will be the percentage of cytosine in that particular DNA sample? Your options are number A 27%, number B 46%, number C 73%, and number D 23%. Again, I will repeat, in a particular human DNA, it has been observed that adenine is present in 27%. So what will be the percentage of the cytosine present in the DNA sample? Number A 27%, number B 46%, number C 73%, and number D 23%. Again, I am giving you some time. This is very easy question. I know all of you can answer this question. So I will wait and then I will check the chat box. In a particular human DNA, it has been observed that adenine is present in 27%. So what will be the percentage of cytosine present in the DNA sample? Just think how adenine binds in the particular DNA, then you will easily you can solve the question. If the percentage of adenine is 27%, what is the percentage of cytosine? Let me check the chat box now. Okay, so I have all of you have answered it. So I have 2 number A and 3 number D. Okay, so A and D are the two different options which I have received as answer. So let me check which of the following will be the correct answer. So the percentage of cytosine present in that particular DNA sample is very good. It's number D 23%. Now let me show you. So the rule of base pairing I have already mentioned, but I am just showing you once more here. So the rule of base pairing in DNA is cytosine and guanine and adenine and thymine. Cytosine and guanine, the number of hydrogen bonding present in cytosine and guanine is 3, and the number of hydrogen bonding present in adenine and thymine is 2. In case of RNA, cytosine binds with guanine, there is no change, but adenine binds with uracil. Okay, so and here also the number of hydrogen bonding is 3 and 2 in the cytosine and guanine pair. It is 3, and adenine and I just let me mention it. So cytosine, there is a 3 hydrogen bond with guanine, and adenine have 2 hydrogen bond with thymine. In case of here also, cytosine have 3 hydrogen bond with guanine, and adenine have 2 hydrogen bond with uracil. Okay, so this is the rule of base pairing, or this is the rule, this is the hydrogen bond. Okay, so now your question says that adenine is present in 27%. So adenine makes a hydrogen bond with thymine. Okay, so since 27% of adenine is present, the percentage of thymine will also be 27%. Both adenine and thymine has to be equal because adenine is binding with the thymine, and there is no just residual adenine on thymine. So this is not possible. The number of adenine will be same with the number of thymine residue. Thus, the remaining two bases, cytosine and guanine, they will consist a total of 100 minus 27 plus 27 means 46%. Okay, now, since adenine and thymine in together they are making around 54%, so the remaining 46% will be belonging to guanine and cytosine. Now guanine and cytosine, they make hydrogen bond with each other, hence they will present in the equal amount. So the number of cytosine, or the percentage of cytosine, will be 46 by 2 is 23%. Okay, just you have to check if adenine is present, is added, there is a in question if adenine is mentioned and you are asking about the cytosine, just you have to do it like a own way, or you can also think it like this, that adenine and cytosine in totally, since adenine and cytosine are in the opposite pair, so they will make around 50% of total, so you can just do it like that also. 50 minus 27 will also give you 23. Okay, so yeah, 23% was the correct answer, and in the chat box I have received this as an answer. So very good. Now I will move to the next problem. So problem number 3 says the position where the tRNA will bind with the ribosome depends on it. Your options are number A, codon sequence, number B, intramolecular hydrogen bonding, number C, anticodon sequence, and number D, hairpin-like structure. Again, I will repeat, the position where the tRNA will bind with the ribosome depends on its number A, codon sequence, number B, intramolecular hydrogen bonding, number C, anticodon sequence, and number D, hairpin-like structure. Where tRNA will bind on ribosome, so the position where it will be binding on the ribosome, it depends on which thing? Codon sequence, intramolecular hydrogen bonding, anticodon sequence, or hairpin-like structure. Let me know in the chat box. A, B, C, or D, which of the following you think is the correct answer of this question? The position where the tRNA will bind with the ribosome depends on it which structure? me check the chat box. Okay, I have received few answers which says it's number C and number A. So I have received two answers. One says it's number C, another says it's number A. So let me check which of the following will be the correct answer. So the position where tRNA will bind with the ribosome depends on it is number C, anticodon sequence. Let me show you first the structure of tRNA. So this is the structure of tRNA. Okay, so there are three different loops of the tRNA. One is known as the acceptor stem, which is a little bit longer. Okay, it has a terminal OH. So this is the 3' position, and this is the acceptor loop. Then we have a T loop. Then we have a variable loop followed by an anticodon loop, and then we have a D loop. Okay, and this position is also known as the anticodon stem. So this is the anticodon loop, which has three particular groups together, which is known as the anticodon sequence, and this anticodon sequence matches the codon sequence of this particular so the amino acid attached here. Okay, and this anticodon sequence matches with the particular codon of the mRNA, and then it will bind to the ribosome. Okay. So how this protein synthesis happen? Let me just show you. So there are three different step of the protein synthesis. One is the initiation step, then the elongation step, and then the termination step. So the first, there are different parts of this or different important structures which are involved in the protein synthesis. One is the tRNA, which brings the amino acid. Then we have the ribosomal unit, one small and one large subunit. We have mRNA, which has different codon sequence, which starts the particular translation process. So the first step of this translation process is the initiation process. So what happens here? Translational complex forms, and tRNA brings the first amino acid in the polypeptide chain to bind to start codon from the mRNA. The start codon is AUG and the stop codon is the UAG. Now there are three different stop codons also, now which are UAG, UGA, and UAA. Okay. So these codons are basically nothing but a three code which consist of three different nucleobases. Okay, and and if a particular codon is codes for a particular amino acid, like here AUG it codes for the methionine, and there are three different codons EUGA, UAG, and UAA, which are known as the stop codon, which do not which indicates where to stop the translation process and release the proton sequence. They do not code for any particular amino acid, they only stop that particular protein synthesis. So in the first step, what happens? Translational complex forms, and tRNA brings the first amino acid to the polypeptide chain to bind to start code for mRNA. Okay. The elongation step is the tRNAs bring the amino acids one by one. Here, tRNA is bringing the amino acid one by one and adding to the polypeptide chain. And the termination step, release factor recognizes the stop codon, and translational complex dissociates and complete polypeptide chain is released. After releasing a complete polypeptide chain, all the components of the particular translational complex like ribosomes, mRNA, tRNA, they are recycled for the next translation cycle. Okay, so that is how the particular translation process. In the examination, if you are asked to write a brief how the protein translation system happen, so that is how you have to answer the questions. Okay, with the figures also. So in the biological questions or in the medicinal questions, the figures you have to put whenever it is applicable. The drawing may not be perfect, but at least it should be indicating some things. Okay. The correct answer was the anticodon sequence. Now I will go to the next problem. So problem number 4 says in prokaryotes the first amino acid in the polypeptide chain is, your options are number A methionine, number B N-methyl methionine, number C N-formyl methionine, and number D all of these. In prokaryotes the first amino acids in the polypeptide chain is number A methionine, number B N-methyl methionine, number C N-formyl methionine, and number D all of these. Again, I will wait for some time. Let me know in the chat box A, B, C or D which of the following is the correct answer. What is the first amino acid which is coded in case of prokaryotes? In prokaryotes, the first amino acid in polypeptide chain is which one? Let me check the chat box. Okay, I have received one answer only which says it's number C. Others are not answering. I will wait for one, no, 2 to 3 more minutes. Okay, I will give you 2 more minutes. Try answering the question. In prokaryotes, the first amino acid in polypeptide chain is which one? Methionine, N-methyl methionine, N-formyl methionine, or all of these? So, any other answer? Okay. Okay. Let me check which of the following will be the correct answer. I have only received one answer, which says it's number C. So the correct answer for problem number 4 is it is number C, N-formyl methionine. This is the structure of the methionine. Okay. So basically, methionine is the first amino acid incorporated into the protein chain in both prokaryotes, here the and eukaryotes. So in the prokaryotes, the methionine is modified as N-formyl methionine. Initiated tRNAs are special tRNAs recognizing the AUG or GUG start codons in prokaryotes and the eukaryotes. The initiator tRNAs differ from one that insert the internal methionine residue. Okay, so AUG is for methionine, and so there is a informal methionine is the first amino acid to be incorporated into the polypeptide chain in prokaryotes. So this is the structure of methionine, which is there also I have mentioned. And informal methionine is nothing. So there is one formal chain is incorporated here. Okay, in with the methionine, one formal chain is incorporated into the informal methionine. Okay. So this is the structure of informal methionine. So this is also so after synthesis, the polypeptide, the formyl group is removed by deformylase. Often N-terminal methionine is also removed by an aminopeptidase as well as one or two additional amino acids. Okay. So correct answer was number C. Now I will go to the next problem. So problem number 5 says which of the following is the following amino acids is involved in the action of the human topoisomerase 2. Your options are number A tyrosine, number B tryptophan, number C serine, and number D proline. Which of the following amino acids is involved in the action of human topoisomerase 2? Number A tyrosine, number B tryptophan, number C serine, and number D proline. Again, let me wait for 2 to 3 minutes. Which of the following amino acids is involved in the action of human topoisomerase 2? Tyrosine, tryptophan, serine and proline. Let me know in the chat box A, B, C or D which of the following you think is the correct answer. Which of the following amino acids is involved in the action of human topoisomerase 2? Let me check the chat box. Okay, so I have received quite a few answers. I think number A is the only option which I have received as the answer of this question. So let me check if number A is the correct one or not. Okay, so the correct answer of problem number 5 is the amino acid which is involved in the action of the human topoisomerase 2 is, it is number A, tyrosine. Yes, it is the correct answer. So topoisomerase 2 is a mammalian enzyme that is crucial for the effective replication of DNA. The enzyme binds to the parts of DNA where two regions of the double helix are near proximity. The enzyme binds to one of these DNA double helices and tyrosine residues are used to nick both these strands of the DNA. This results in temporary covalent bond between the enzyme and resulting 5' end of each strand, thus stabilizing the DNA. The strands are now pulled in the opposite direction to form a gap through which the intact DNA region can be passed. The enzyme then reseals and strand then departs. So basically, what is the role of the topoisomerase 2? The topoisomerase 2 here, so this topo catalyzes the crossover of the DNA strand. Then the same enzyme binds covalently into with each strand DNA strand. So this is one DNA and this is another DNA strand. So this is in case of the when there is a DNA replication. So there is a supercoiling of the two different DNA strand. So how this supercoiling is relaxed by this topoisomerase? So it has a tyrosine amino acid. Okay, and with it it binds with the to the strands of the DNA, and when it and then it breaks the two strands. Okay, and in the passage and creates a gap from where the intact DNA can pass. So as you can see here is one DNA strand, and topoisomerase 2 is binding with each strand and nicking it at any position, and then a gap is created, in which gap the intact DNA strand it can pass through, and after doing it, this topoisomerase, this enzyme, it reseals the nicks which is it has created, and then it leaves. Okay, so so that is how this DNA topoisomerase 2 works. In case of the topoisomerase 2, so how this tyrosine residue is placed. So this is the nucleus 2 nucleosides. I have already shown you how two nucleosides are bonding each other with each other through a phosphate linkage. The tyrosine, it has one phenolic OH group. This phenolic OH group participates in the nucleophilic attack with this phosphate moiety, and that is how it binds with this one phosphate and creates a break in between a DNA chain. Okay. So yeah, tyrosine was the correct answer. Now I will go to the next problem. So problem number 6 says the type of topoisomerase which generally relaxes the DNA by removing negative supercoiling is, your options are number A type 1, number B type 2, number C type 3 and number D type 4. So the type of topoisomerases which generally relaxes DNA by removing the negative supercoiling is which one? Type 1, 2, 3 or 4. I am giving you 2 minutes, then I will check the chat box. Let me know there which will be the correct answer. The type of topoisomerases which generally relaxes DNA by removing negative supercoiling is type 1, type 2, type 3 or type 4. A, B, C or D. So there are 4 different options. You have to answer it in the chat box. A, B, C or D. Which of the following you think is the correct answer? So the type of topoisomerases which generally relaxes the DNA by removing negative supercoiling is, let me check the chat box. Okay, I have received 2 answers. Both are saying it is number A. Let me check if number A is the correct answer or not. Yes, number A is the correct one. Type 1 topoisomerase generally relaxes the DNA by removing the negative supercoiling. So what is the supercoiling of DNA? So the topology, so there is a two type of supercoiling. One is right-handed supercoiling, which is known as the negative supercoiling or underwinding, and there is a left-handed supercoiling, which is known as which is a positive supercoiling. So relaxed states of the DNA do not have any bends. The DNA must be constrained, plasmid DNA or by proteins. Unraveling the DNA at one position changes the superhelicity. Topology only defined for the continuous deformation, no strain breakage. So topoisomerase 1 versus topoisomerase 2. So majorly there are two topoisomerases 1 and 2 are present. So topoisomerases 1 are which cut the one of the two strands of double helix and relax the strand and reanneal the strand. Topoisomerase 2, the enzymes which cut both strands of the DNA DNA helix simultaneously in order to manage the DNA tangles and supercoils. The type 1 generates single strand breaks, type 2 generates double strand breaks. Type 1 occurs in eukaryotes, type 2 occurs in both eukaryotes and prokaryotes. Type 1 does not require ATP hydrolysis, type 2 require ATP hydrolysis. Type 1 there is 3 subclasses of type 1 topoisomerase or type 1 topoisomerase, that is type 1A, type 1B and type 1C, and 2 subclasses of type 2 topoisomerase are there, type 2A and type 2B. Okay. So type 1 is the correct answer. I will go to the next problem. Problem number 7 says, till now we were discussing about the nucleic acids part. So now we will switch to the enzyme inhibition topics. Okay. So the question number 7, it says that which of the following diagrams best describes the equilibrium associated with a competitive inhibitor? Your options are here. And E suggests the enzyme, S denotes the substrate, and P denotes the product, and I denotes the inhibitor. Okay. Number A is this one. Where E plus S in equilibrium with ES in equilibrium with E plus P. ES is in equilibrium with EIS and E is equilibrium with EI. Okay, so this is the number B, this is the number C and number D. Which of the following is the correct equilibrium associated with a competitive inhibitor? E denotes enzyme, S denotes substrate, P denotes product, and I denotes the inhibitor. A, B, or C, or D, you have to answer. Let me know which of the following is the correct answer. I will give you some time. Look at the all the equilibrium carefully. Just think what does a competitive inhibitor do, and then let me know in the chat box. Which of the following diagrams best describes the equilibrium associated with a competitive inhibitor? E, S, P, or I? E in denotes enzyme, S denotes substrate, P denotes product, I denotes inhibitor. A, B, C, or D both are denoting a particular equilibrium. So you have to find out which of the following is the correct answer for a competitive inhibitor. Let me check the chat box. So I have received, I think, only one answer for this question, which says it's number D. Let me check if number D is the correct answer for this problem. So the diagram which best describes the equilibrium associated with a competitive inhibitor is number C. Okay, number C is the correct answer. So here the enzyme is in equilibrium with the inhibitor, which forms a enzyme inhibitor complex, just same as the enzyme substrate complex. So what is the competitive inhibition? What is competitive inhibition of enzymes? So the competitive inhibitor has roughly the same shape as the substrate, so it can fit in the active site of the enzyme. So we all know enzyme has an active site which has a particular size and particular shape, and the shape of and the size of this particular enzyme's active site is such that only the substrate molecule can bind it. But competitive inhibitor is a molecule or a particular inhibitor. It is an entity which binds, which has the roughly same shape and same size of the particular substrate, and it can bind into the active site of the enzyme. Okay, the competitive inhibitor is able to fit at the active site. When it is there, the substrate is unable to bind to the enzyme and the desired reaction does not occur. The inhibitor and the substrate both compete for the active site. That is why it is known as the competitive inhibitor. The competitive inhibitor is often a product of the enzyme catalyzed reaction. When there are a lot of product is made, the product would compete with the substrate for the enzyme's active site, showing the reaction down. Okay. So here you can see the enzyme and the inhibitor and the substrate both are competing to form an enzyme substrate complex and enzyme inhibitor complex. Okay, that is why this is the equilibrium, which is suggesting that the enzyme can form the enzyme substrate complex with the substrate. Also, it can form the enzyme inhibitor complex with the enzyme. Okay, because the competitive inhibitor has the same shape of the substrate. Okay, they compete for the same active site of an enzyme. So what is the normal enzyme? The maximum rate of the reaction can be reached when the line plateaus. All enzymes are occupied with the line plateaus. So when all the enzymes are occupied with the substrate. Competitive inhibitor. Remember, the competitive inhibitor competes for the active site with the substrate. When the substrate concentration exceeds the inhibitor concentration, the reaction will proceed and maximum rate of the reaction can be achieved. Non-competitive inhibitor also there. These inhibitor don't compete or for the active site. They attach to an alternative site of the enzyme, changing the shape of the active site, preventing the substrate from binding. Non-competitive inhibitor cannot be overcome by increasing the substrate concentration. Therefore, maximum rate of the reaction will not be achieved regardless of the substrate concentration. This inhibition lowers the amount of the usable enzymes. Okay, so one way to attain the highest rate of the reaction in presence of competitive inhibitor is increasing the substrate concentration. So if there is a more substrate than the inhibitor molecule, the most of the active sites will be occupied by the substrate molecules itself, not the inhibitor molecules. Okay, so that is one way to attain the maximum rate in case of the competitive inhibition, but it is not possible in case of the non-competitive inhibition. Okay, so number 3 was the correct equilibrium which is associated with the competitive inhibitor. Now next problem I will show you. Problem number 8 says, what is the mechanism of action of the cyanide poisons? Your options are number A, competitive inhibition, number B, non-competitive inhibition, number C, uncompetitive inhibition, number D, irreversible inhibition. What is the mechanism of action of the cyanide poisons? Competitive inhibition, non-competitive inhibition, uncompetitive inhibition, or irreversible inhibition? I will wait for some time and then I will show you the answer. What is the mechanism of action of cyanide poisons? Competitive, non-competitive, uncompetitive, irreversible. So what is the mechanism of action of cyanide poisons? Competitive inhibition, non-competitive inhibition, uncompetitive inhibition, irreversible inhibition. Let me check the chat box. Okay, I have few answers, I think. Number B, number A, number B, number D. So I have three different options as answer. Number B, A, B and D. Three different options I have received as answer. Let me check which of the following is the correct one. So the correct answer, the mechanism of action of the cyanide poisons is, it is number B, non-competitive inhibition. So what is non-competitive inhibition? The non-competitive inhibition does not have a structural similarity with the substrate, but it binds with the free enzyme and the enzyme substrate complex. Okay, so the non-competitive inhibitor, it does not compete with the active site of the enzyme, but it binds to another position of the particular enzyme, and also it can bind with the enzyme substrate complex. The simplified equation of the steady state kinetics of the non-competitive inhibition is this one. Okay, the inhibitor can bind with inhibitor enzyme go to form an enzyme inhibitor complex. It can bind with the enzyme substrate complex to bind a enzyme substrate inhibitor complex. Okay, the presence of the substrate has no influence on the ability of the non-competitive inhibitor to bind an enzyme or vice versa. Its binding alters the conformation of the enzyme and reduces its catalytic activity due to the changes in the nature of the catalytic groups at the active site. EI and ESI complexes are non-productive. Okay, so this EI and ESI are the non-productive. On increasing substrate to a saturated concentration does not reverse the enzyme, leading to unaltered Km, but also reduced Vmax. Reversal of the enzyme inhibition requires a specific treatment like dialysis or pH adjustment. Examples of the non-competitive inhibitors are mostly poisons. Cyanide and azide inhibit the enzymes with iron and copper as the component of the active site of the prosthetic group, that is, for example, in cytochrome C oxidase, cyanide binds with the iron 3 in the heme of the cytochrome AA3 component of the cytochrome C oxidase and prevents the electron transport to oxygen. Mitochondrial respiration and energy production cease and cell death occurs. This central nervous system is the primary target of the cyanide toxicity. Acute inhalation of the high concentration of cyanide, like smoke inhalation during the fire or automobile exhaust, provokes the brief central nervous system stimulation rapidly followed by convulsion, coma and death. Acute exposure of the lower amounts can cause lightheadedness, breathlessness, dizziness, numbness and headaches. Cyanide is present in air as hydrogen cyanide, in soil and water as cyanide salts, and in the foods as cyanoglycosides. Okay. So cyanide poisons are example of non-competitive inhibitor. What does this non-competitive inhibitor do? They bind with free enzyme or enzyme substrate complex to form a non-productive ESI complex. Okay, and increasing the substrate concentration will not reverse this kind of inhibitions. Okay. Now I will go to the next problem. Problem number 9 says, which one of the following inhibition is called the mechanism-based enzyme inhibition? Your options are number A, non-competitive inhibitor, number B, non-competitive inhibitor, number C, suicide inhibition, and number D, active site directed irreversible inhibition. Which one of the following inhibition is called mechanism-based enzyme inhibition? Competitive inhibition, non-competitive inhibition, suicide inhibition, active site directed irreversible inhibition. Which one of the following is called mechanism-based enzyme inhibition? In the chat box, let me know A, B, C or D, which of the following is the correct answer for problem number 9. Which one of the following inhibition is called mechanism-based enzyme inhibition? Let me check the chat box. Okay, I have quite a few answers. Number D, number C. So I have two different options, C and D. Let me check which of the following is the correct answer. So the inhibition which is called mechanism-based inhibition is number C, in suicide inhibition. Okay. So suicide inhibition, it is known as the mechanism-based inactivation. The inhibitor makes the use of the enzyme's own reaction mechanism to inactivate it. In suicide inhibition, the structural analog is converted to more effective inhibitor with the help of the enzyme to be inhibited. Now, for example, ornithine decarboxylase catalyzes the conversion of ornithine to putrescine, which is necessary for the polyamine synthesis. When the ODC is in trypsinoma is inhibited, the multiplication of the parasite is arrested. Therefore, inhibitors of this ODC enzyme such as difluoromethyl ornithine have been found to be effective against trypsinomyces. A similar mechanism is observed in case of the allopurinol, which is oxidized by xanthine oxidase to alloxanthine, that is strong inhibitor of the xanthine oxidase. The inflammatory action of aspirin is also based on the suicide inhibition. Arachidonic acid is converted to prostaglandin by the enzyme cyclooxygenase. Aspirin acetylates the serine residue in the active center of cyclooxygenase. Thus, prostaglandin synthesis is inhibited, so inflammation is subsided. Okay. So basically, the suicide inhibitors, they use the enzymes or mechanism to inactivate it. Okay, that is why it is called the suicide, because it is the enzyme itself, its mechanism is killing its action. Okay, that is why it is called the suicide inhibition. What is problem number 10? It says what is the unit of the turnover number in the enzyme? Your options are number A, concentration per time, number B, time inverse, number C, concentration inverse, and number D, time per concentration. What is the unit of the turnover number of the enzyme? Turnover number is a very important parameter of enzyme activity. Now you have to identify what is the unit of this turnover number of the enzyme. Your options are number A, concentration per time, number B, time inverse, number C, concentration inverse, and number D, time per concentration. Let me know in the chat box. I will give you some time. A, B, C or D. Which of the following you think is the correct unit of the turnover number of an enzyme? Turnover number is a very important parameter of an enzyme. What is the unit of the turnover number of an enzyme? Concentration per time, time inverse, concentration inverse, or time per concentration. Let me check the chat box. Okay, I have quite a few answers. Number A, number C, number B. So I have three different options: A, C, and B. These three different options I have received as answer for this problem number 10. Okay, let me check which of the following is the correct answer. So the correct unit of turnover number of the enzyme is, it is number B, time inverse. So what is the turnover number of an enzyme? Turnover number is also termed as kcat, is defined as the maximum number of chemical conversions of the substrate molecules per second that is that a single catalytic site will execute for a given enzyme concentration. Again, I am saying turnover number is also termed as kcat, is defined as the maximum number of chemical conversions of the substrate molecules per second that is that a single catalytic site will execute for a given enzyme concentration. Okay, so kcat equal to Vmax by enzyme. This is the by ET, which is the enzyme particular enzyme concentration. For example, carbonic anhydrase has a turnover number of 4 lakh to 6 lakh second inverse, which means each carbonic anhydrase enzyme molecule can produce up to 6 lakh molecules of the product, that is bicarbonate ion, per second. Okay, so this per second or per time or time inverse is the unit of the turnover number. Okay, now I will go to the next problem. Problem number 11 says the drug that inhibits the initiation step of the translation process is, your options are number A, atorvastatin, number B, nilotinib, number C, streptomycin, and number D, artemisinin. Which of the following drug inhibits the initiation step of the translation process? Already I have mentioned the translation process, how the translation process happen. If you remember, there are three different steps of the translation process. One is the initiation step. Second, first is initiation, second is the elongation, third is the termination step. So the initiation step of the translation process is inhibited by which drug? Your options are number A, atorvastatin, number B, nilotinib, number C, streptomycin, and number D, artemisinin. Which of the following drug inhibits the initiation step of the translation process? The drug that inhibits the initiation step of the translation process is which one? Let me check the chat box. Okay, I have got two options, number C and number B. Okay, let me check which of the following is the correct answer, number C or number B. So, the drug that inhibits the initiation step of translation process is, it is number C, streptomycin. So what is the mechanism of action of streptomycin? So streptomycin binds to the 30S ribosomal subunit. Streptomycin inhibits the S12 and 16S rRNA of 30S small ribosomal subunit encoded by the genes RPSL and RRS. Okay, and wrong pairing of the codon, anticodon reaction, and misreading the genetic code blocks the translation process. The mechanism of resistance is, the resistance is due to the mutation. So these drugs are not that all the time this drug will work. Sometimes there is a resistance of the drug resistance, and what is the reason of the resistance? Resistance due to the mutation of the RPSL and RRS genes. Okay, so this is the mechanism of action of streptomycin. So this is the protein synthesis inhibition. So there are different drugs or different chemical entity which inhibits the protein synthesis, especially as antibacterial agents. They are very good antibacterial agents. Okay, like streptomycin changes the shape of the 30S ribosomal subunit, causing code on mRNA to be read incorrectly. Okay, like this, it is bending this particular code so that the code there is no codon anticodon pairing happens here. Then tetracyclines are also antibacterial agents, very important antibacterial agents, which also inhibits the protein synthesis of the bacteria. These tetracyclines inhibit the attachment of tRNA and mRNA ribosomal complex. Then there are chloramphenicol, which binds to the 50S portion and inhibits the formation of the peptide bond. Okay, so there are different chemical agents or different drug moieties which bind to different portion of the translational complex to inhibit the protein synthesis, and these drugs are very important antibacterial agents also. Okay, because they inhibit the bacterial protein synthesis. So now I will go to the next problem. Problem number 12 says fetal hemoglobin has a higher binding activity for oxygen than does the adult hemoglobin. In comparison to the adult hemoglobin, oxyhemoglobin dissociation curve, the fetal oxyhemoglobin dissociation curve will be: number A, shifted to the left and display lower Km. Number B, shifted to the right and display lower Km. Number C, shifted to the right, display higher Km. And number D, shifted to the left to display higher Km. Fetal hemoglobin has a higher binding activity of our oxygen than the adult hemoglobin. In comparison to the adult oxyhemoglobin dissociation curve, the fetal oxyhemoglobin dissociation curve will be: number A, shifted to the left and display lower Km. Number B, shifted to the right and display lower Km. Number C, shifted to the right and display higher Km. Number D, shifted to the left and display higher Km. Read all the options and question very carefully, and then let me know in the chat box A, B, C or D which of the following will be the correct answer. So let me check the chat box now. Okay, I have received an answer which says it is number C. One is saying it is the number A. So two options I have received as answers, number A and C. Let me check which of the following is the correct one. So the correct answer will be, it is number A, shifted to the left and display lower Km. So adult hemoglobin, it has 2 beta chain and 2 alpha chain, and fetal hemoglobin has 2 alpha chain and 2 gamma chain. Okay, the polypeptide gamma chains enable to fetus to have the greater affinity for oxygen. It can be ensured the oxygen can be delivered to the organs to developing fetus. So fetal hemoglobin has a higher affinity of oxygen at all partial pressures. Okay, so this is the curve is for fetal hemoglobin, and this curve is for the adult hemoglobin. This ensures the oxygen is transferred to the fetus from the maternal blood across the placenta. The partial pressure of fetal tissues is very low. Due to this, the fetal hemoglobin metabolic rate, due to the high metabolic rate of the associated with the fetal growth rate. Although fetal hemoglobin has a higher affinity of oxygen, in such low partial pressure environment of the fetal is unload the oxygen to the readily. At birth, fetal hemoglobin is replaced by the adult type hemoglobin. Okay, so this curve is shifting to the left, and the Km is the displaying lower Km. Okay, so Km I already have mentioned in the last class. It is the substrate concentration at the 50% of the the rate of the enzyme. Okay, so as you can see, if we draw a perpendicular from this 50% of this percentage of saturate of oxygen, and then draw a perpendicular on the x-axis, it shows that the Km shows the Km. So this is the Km for the fetal hemoglobin, and this is the Km for the adult hemoglobin, which shows that the Km is lower in case of the fetal hemoglobin. So fetal hemoglobin has a greater affinity toward the oxygen rather than the adult hemoglobin. This ensures that from mother's tissues, the fetal tissues, they get the oxygen, and at the birth the adult hemoglobins are replaced with the fetal hemoglobin. Adult hemoglobins are the adult hemoglobin, they replace the fetal hemoglobin in the growing in the baby, okay, after its birth. Okay, so option A was the correct now. Now problem number 13 says, what is the mechanism of action of early state A anti-obesity drug? Your options are number A competitive inhibition, number B non-competitive inhibition, number C uncompetitive inhibition, number D irreversible inhibition. What is the mechanism of action of early state and anti-obesity drug? Competitive inhibition, non-competitive inhibition, uncompetitive inhibition, or irreversible inhibition? What is the mechanism of action of early state? Let me know in the chat box, A, B, C, or D, which of the following you think is the correct answer. What is the mechanism of action of early state? Competitive inhibition, non-competitive inhibition, uncompetitive inhibition, and irreversible inhibition. Let me check the chat box. Okay. I have received two answers which are saying it is number D. Let me check if number D is the correct answer. So, The mechanism of action for the of orlistat, which is an anti-obesity drug, is, yes, it is number D, irreversible inhibition. So irreversible inhibition for the treatment of obesity. Fat in the diet is composed mainly of the triglycerides, which are digested in the small intestine to fatty acids and to monoglyceride. The digestion products are then absorbed and act as a building blocks for the fat biosynthesis in the body. The enzyme pancreatic lipase is responsible for catalyzing the digestion of the fat. So inhibition of the enzyme will result in reduced absorption of glycerides and fatty acids from the gut. Consequently, less fat will be synthesized in the body. Orlistat is an anti-obesity drug which acts as an irreversible inhibitor of the pancreatic lipase as a result of the presence of the electrophilic four-membered lactone group. This acylates the serine residue of the active site, which is a part of the catalytic triad of serine, histidine, and aspartic acid. Okay, so this is in this case of this pancreatic lipase, there is a catalytic triad. Remember, I have in the last class I have mentioned that there is a catalytic triad in presence in the enzyme chymotrypsin, which is also serine, histidine, and aspartic acid. Here also there is a catalytic triad of serine, histidine, and aspartic acid is present. Okay, so from this this orlistat, this is the structure of orlistat. What is do? It has a four-member lactone ring. So now what this serine residue, whenever this orlistat is closer by this pancreatic lipase, this serine residue, it acts as a nucleophile and opens up this lactone ring to form this kind of moiety. This moiety is binding with this serine residue, and now the serine this serine hydroxyl group cannot act as a nucleophile anymore, and this inhibits the activity of this enzyme pancreatic lipase. So this is a example of irreversible inhibition. Okay. Irreversible enzyme inhibitors can form covalent bond to the key amino acid of the active site and permanently block the affected enzyme. The most effective irreversible inhibitors are those that contain an electrophilic functional group that capable of reacting to the nucleophilic group of the amino acid side chain. Invariably, the amino acid affected is either serine or cysteine because these amino acids are often present in active sites and contain nucleophilic group like OH and SH respectively that are involved in the mechanism of many enzyme catalyzed reactions. Electrophilic functional groups used in reversible inhibitors include alkyl halide, epoxide, alpha beta unsaturated ketone, or strained lactones or lactam. The highly toxic nerve agents contain electrophilic fluorophosphate groups are irreversible inhibitors of the mammalian enzyme. Irreversible enzyme inhibitors are not competitive inhibitor. Increasing the concentration of substrate will not reverse the action of the inhibitors since they cannot be replaced from the active site. This can cause problem if buildup of the particular substrate leads to the toxic side effects. For example, monoamine oxidase inhibitors of myosin block the metabolism of noradrenaline and have antidepressant activity. Unfortunately, the metabolism of substrate other than the noradrenaline is also inhibited, leading to the buildup of these compounds and serious side effects. More modern myoins are designed to be the reversible inhibitors in order to avoid this problem. Okay. So yeah, option D was the correct answer. Now I will go to the next problem. Problem number 14 says inhibition of cytochrome P450 by thiolinic acid is an example of dash inhibition. Your options are competitive, non-competitive, number A competitive inhibition, number B non-competitive inhibition, number C suicide inhibition, number D active site directed irreversible inhibition. Inhibition of cytochrome P450 by thiolinic acid is an example of which kind of inhibition? Number A competitive inhibition, number B non-competitive inhibition, number C suicide inhibition, number D active site directed irreversible inhibition. Inhibition of cytochrome P450 by thiolinic acid is an example of which inhibition? Let me check the chat box. Okay, so I have few answers. I think I have three answers. All the three answers are saying it's number C. Let me check if number C is the correct answer or not. So the correct answer is yes, it's number C. Very good. Suicide inhibition. So, suicide substrates are agents which are converted to highly reactive species when they undergo an enzyme catalyzed reaction. They form covalent bond with the enzyme, inhibit it irreversibly. In some cases, it can cause toxicity. For example, diuretic agent tylenic acid had been withdrawn from the market because it was found to act as a suicide substrate for the cytochrome P450 involved in the drug metabolism. Unfortunately, the metabolic reaction carried out by these enzymes converted tylenic acid to the thiophene sulfoxide, which is proved highly electrophilic. This encouraged a Michael reaction leading to alkylation of the thiol group in the enzyme active site. Loss of water from this thiophene sulfoxide restored the thiophene ring and resulted in the formation of the covalent link to the enzyme, thus inhibiting the enzyme irreversibly. This is the structure of tylenic acid. It has a thiophene moiety. Okay, here, and in the cytochrome P450 we have a nucleophilic thiol group here from this cysteine residue. Okay, so for oxidation of the cytochrome P450, what does it do? Basically, oxidation of this thiophene group, it makes this group electrophilic and accelerates the nucleophilic attack by this thiophene moiety. This is and then there is a covalent linkage of this part with this after alkylation, and it's a Michael addition, and eventually it forms enzyme alkylated and inhibit. This enzyme gets alkylated and inhibited by this tylenic acid. Okay. So this is the mechanism of action which also involves a Michael react type of reaction. Okay. So remember, tylenic acid is an inhibitor, suicide inhibitor of cytochrome P450 enzyme. Problem number 15 says which inhibition has apparent Km value as Km of the normal enzyme catalyst reaction. Your options are number A competitive, number B non-competitive, number C uncompetitive, number D mixed. Which inhibition has apparent Km value as Km of the normal enzyme catalyzed reaction? Apparent Km means in case of normal enzyme catalyzed reaction, the Michaelis-Menten constant is known as Km, but in case of enzyme inhibitions, okay, the particular Km is known as the apparent Km, which is not the same Km as the normal enzyme catalyzed reaction. Which inhibition has apparent Km value as Km of the normal enzyme catalyzed reaction? Competitive, non-competitive, uncompetitive, or mixed? Let me know in the chat box, A, B, C, or D, which of the following will be the correct answer. Which inhibition has apparent Km value as Km of same as the Km value of normal enzyme catalyzed reaction? Okay, let me check the chat box now. Okay, I have got three answers. Okay, so, three answers are saying it's number B, one is number C. Okay, let me check B or C. Which of the following is the correct option? So the inhibition that has apparent Km value same as the normal Km of normal enzyme catalyzed reaction is number B. Okay, non-competitive inhibition. This table you have to remember. I will suggest to you to remember this table. Okay, so type of inhibition, the Vmax, apparent Vmax by Km, apparent Km and apparent Km. So this is the table for no competitive inhibition. Apparent Vmax is same as the Vmax of the normal enzyme catalyst reaction. The apparent Vmax by apparent Km is this one, and the apparent Km is this one. Km into 1 plus concentration of inhibitor by Ki. Okay, in case of the mixed inhibition, this is the value of the apparent Vmax, the Vmax divided by 1 plus concentration of I divided by concentration Ki. This is the value of the Vmax apparent and Km apparent, and this is the value of Km apparent. In case of non-competitive inhibitor, this is the value, this is the particular Vmax apparent, this is the Vmax by Km, and this is the Km apparent. In case of uncompetitive inhibitor, these are the value in case of this particular Vmax by Km remains same as the apparent Vmax by Km. Okay, so how all these values are coming, this is the derivation is not in your syllabus, but if you want me to show you how these things are coming, so I will show you this in the summary class. Okay, at the end of our all 12 courses we will have a summary class. There I will discuss the topics which were not in detail in your course, but which sometimes we require, so that things I will cover up in the summary class. Okay, so for now remember this table and just try to memorize the table, and also with the equilibrium which I have shown you in the first slide, in the earlier slides. Okay, the equilibrium of a particular enzyme catalyst reaction inhibitions, different inhibition, and remember the table how this values are coming. I will show you in the summary class, which is at the end of our particular course. Okay, now I will go to the next problem. Problem number 16 says, which inhibition study deals with the decrease of the value of both Vmax and Km with the increase of the inhibitor concentration? Your options are number A competitive, number B non-competitive, number C uncompetitive, number D suicide. Which inhibition study deals with the decrease in the value of both Vmax and Km with the increase of the inhibitor concentration? Your options are number A is competitive, then non-competitive, number C is uncompetitive, number D is suicide inhibition. Both in decrease of the value of Vmax and Km. I will give you some time. Think and let me know in the chat box. A, B, C or D, which of the following is the correct answer? Which inhibition study deals with the decrease of the value in both Vmax and Km with the increase of inhibitor concentration? Let me check the chat box. Okay, I have received 3 answers and all the 3 are saying it is number D. Okay, let me check. So the correct answer is number C, uncompetitive inhibition. Okay, uncompetitive inhibition, they deals with the decrease of both Vmax and Km with the increase of the inhibitor concentration. So these are the Lineweaver-Burke plot of the inhibition. In the last class I have discussed what is the Lineweaver-Burke plot and how to derive this Lineweaver-Burke plot from the Michaelis-Menten plot. Okay. So here in the Lineweaver-Burke plot we plot the 1 by V in the y-axis and 1 by S in the x-axis. Okay. So this is the Lineweaver plot for the inhibition. This is for the competitive inhibition. This is for the uncompetitive inhibition, and this is for the your non-competitive inhibition. So in case of the competitive inhibition, Km increased and Vmax, as you can see, what is the, so in case of Lineweaver-Burke plot, what is the slope and intercept? The slope value is Km by Vmax, okay, and intercept is 1 by Vmax. So as you can see, in presence of inhibitor, this red one, the slope is increasing, okay, but the intercept is remaining the same. So since the intercept remaining the same, the Vmax remains unaffected. But since this particular slope is increasing, means the 1 by Km is increasing, means Km is decreasing. Okay, so sorry, I am sorry. The slope is Km by Vmax. Okay, since the slope, sorry, I just made a mistake. So slope is Km by Vmax. Since the slope is increasing and Vmax is remaining unaffected, it means that Km is increasing in case of the competitive inhibition. In case of the uncompetitive inhibition, as you can see, both the slopes are the same, but the value of intercept is increasing, okay, in presence of inhibitor, means 1 by Vmax is increasing, means Vmax is decreasing, and since slope means Km by Vmax is remaining the same, it means that the Km will also have to decrease to keep the slope same. Okay, then also in case of uncompetitive inhibition, Km and Vmax both are decreasing, and in case of non-competitive inhibition, as you can see, Km is remaining unaffected since 1 minus 1 by Km is remaining the same. So Km is remaining unaffected, but the slope is increasing. That is why Vmax is reducing. Okay. So here I will again repeat, 1 by Vmax is remaining unaffected, so Vmax is unaffected, but the slope is increasing. Slope means the Km by Vmax, okay, it remains unaffected. Since Km is remaining unaffected, means the Km is increasing, okay. In case of the uncompetitive, as you can see, slope is increasing, but 1 by Vmax is decreasing, means Vmax is increasing, means Vmax is decreasing, and slope is also, since slope is increasing, this Km is also decreasing. But here Km is unaffected, okay, Km is unaffected, but Vmax is reduced since and the slope, as you can see, the slope is also increasing. So if Vmax is reduced and slope is increasing, it means that Km has to remain unaffected, okay. So this is all the plots are associated with all the Lineweaver plots associated with the enzyme inhibition. This is very important. I will suggest you to remember how the plots look, and from that you can easily answer this type of question. But this is very important. Okay. Now problem number 17, it is a mathematical problem. It says that the turnover number of chymotrypsin is 100 per second, and for the DNA polymerase it is 15 per second. The velocity of the reaction catalyzed by both enzymes at saturating substrate level could be made equal if dash times more DNA polymerase than chymotrypsin were used. Fill in the blanks. Again, I will repeat, turnover number for the chymotrypsin is 100 per second, means 100 second inverse, and for the DNA polymerase it is 15 second inverse. The velocities of the reactions catalyzed by both enzymes at saturating substrate concentration can be made equal if dash times more DNA polymerase than chymotrypsin were used. You have to fill in the blanks. Your options are number A 1500, number B 0.15, number C 67, and number D 6.7. The turnover number for chymotrypsin is 100 second inverse, DNA polymerase is 15 second inverse. So if dash times more DNA polymerase than chymotrypsin were used, then the velocity of the reaction catalyzed by both enzyme at saturating substrate levels could be made equal. What will be the correct answer? Try to do it. If you have a pen and paper, try to read the question very carefully. Think what is the turnover number, which I have mentioned already. I will give you some time to solve the question. I will wait for 2 to 3 minutes, then I will check the answers given by you, and then I will show you how to solve this type of question. Let me check the chat box. Okay, I have got two answers which are say it is number D. Okay, let me check if number D is the correct answer or not. Yes, very good. Number D is the correct answer. So what is the turnover number? I have already mentioned turnover number or kcat is defined as the number of product molecules formed by each enzyme active site per second. So Vm equal to kcat into the enzyme substrate enzyme concentration total. Okay, so think kcat as the frequency of the catalysis, that is the number of catalysis cycle per 7. The value of kcat is the measure of the catalytic efficiency. Fastest the enzyme have highest kcat value, so only kcat is a true constant. Its value only depends on the enzyme's catalytic power. Note that Vm cannot be a true constant because the value of Vm depends on the enzyme concentration. So now Michaelis-Menten equation is V0 equal to K2 into S0 by into E0 divided by S0 plus Km. At a high substrate concentration, S0 is much greater than Km. So the equation looks like this: rate equal to K2 into E0 equal to Vmax. Okay, that is the rate attains the maximum value and the reaction is of zero order with respect to the substrate concentration. The K2 is called the turnover number now of the enzyme that represents the number of molecule converted to product unit time by 1 unit of 1 molecule of enzyme. So let's assume Xc is the strength of the molarity of the chymotrypsin enzyme and Xd is the enzyme of the DNA polymerase, concentration of the DNA polymerase. The Vmax of chymotrypsin equal to 100, which is the turnover number, into Xc, and Vmax of DNA polymerase equal to turnover number of DNA polymerase, which is 15 second inverse into Xd. Now, if the velocity of the reaction of the both the reaction catalyzed by the enzymes at saturated substrate level could be made equal if 100 into Kx Xc equal to 15 into Xd, means Xd divided by Xc is equal to 100 divided by 15 equal to 6.7. That is, the concentration of DNA polymerase has to be 6.7 times more than the chymotrypsin. Okay. So if 6.7 times more DNA polymerase is present than chymotrypsin, then only this both the reaction rates at the highest velocity can be made equal. So, and option number D is correct. I hope you have understood this problem. When I will share this PPT, try to solve it by your own one more time. I think that then it will be more clear. Okay. There's nothing that you have to remember, that Vmax equal to K2 into E0, where K2 is the turnover number and E0 is the enzyme concentration. Okay. So then we are assuming that Xc is the enzyme concentration of the chymotrypsin and Xd is that of the DNA polymerase. K2 values are already given to us. The turnover number of chymotrypsin is 100 per second and the DNA polymerase is 15 per second. And then the velocities will be make equal if 100 into Xc equal to 15 into Xd, means Xd by Xc equal to 6.7, means DNA polymerase has to have the concentration of DNA polymerase has to be 6.7 times higher than that of the chymotrypsin. Okay. Now let me go to the next problem. Problem number 18 said, consider the plot of 1 by V0 by S of the Lineweaver-Burke plot. The result obtained from the graph is the intercept value is 0.2 micromolar minute inverse and the slope is 7 into 10 to the power 5 minute. What is the Km of the reaction? Your options are number A 35 into 10 to the power 5 micromolar, number B 0.258 into 10 to the power 5 molar, number D 7 into 10 to the power 6 micromolar, and number D 0.001 molar. 0.011 molar. So Lineweaver-Burke plot I have already mentioned in the last to last question, and I have already mentioned what is the plot and what is the slope and intercept for the Lineweaver-Burke plot of enzyme catalysis. Only you have to put the values there and you will find out your answer. I will again give you 2 to 3 minutes. Try to solve the question and let me know the answer in the chat box. The plot of Lineweaver-Burke, where y-axis is 1 by V0 and in the x-axis we have 1 by S. The value of intercept is given and value of slope is given. You have to find out what is the Km of the particular reaction. Let me check the chat box now. Okay, I have one answer which says it's number C. Okay, let me check if number C is the correct answer or not. So the value of Km will be, it's number A. Okay, fine. Let me show you how to solve this question. So I have already mentioned the Lineweaver-Burke plot or double reciprocal plot is a common way of illustrating kinetic data. This is produced by taking the reciprocal of both sides of Michaelis-Menten equation, that is a linear form of Michaelis-Menten equation, and it produces a straight line with the equation y equal to mx plus c. The y-intercept is equivalent to 1 by Vmax and the x-intercept is equal to minus of the graph x is equivalent to representing minus 1 by km. The double reciprocal form of Michaelis-Menten equation is this one, 1 by v equal to km by v max into 1 by s plus 1 by v max. Okay, so if you compare it with the y equal to mx plus c, so what we will have, the y equal to mx plus c, we will give you this one, y equal to mx plus c. C means the intercept, so this is the intercept value 1 by Vmax and this is the particular Km by Vmax is your slope. Okay, the Km by Vmax is equal to your slope and intercept, okay, sorry. The Km by Vmax is your slope and 1 by Vmax is your intercept. Now let's go back to the our particular option. Okay, so here the intercept value is given 0.2. Intercept means 1 by Vmax value is given as 0.2 micromolar minute inverse and the slope Km by Vmax is given as 7 into 10 to the power 5 minutes. So now from here we can solve it. The Lineweaver-Burke plot is associated with 1 by V0 equal to 1 by Vmax as I have mentioned already. So now intercept is 1 by Vmax, slope is Km by Vmax. For the question, it is given that the intercept or 1 by Vmax equal to 0.2 micromolar minute inverse. So Vmax will be 5 micromolar into minute inverse, just we have to make it reciprocal, and slope is given by Km by Vmax. Okay, so Km will be slope into Km will be slope into Vmax equal to, so slope is given 7 into 10 to the power 10 to the power 5 minute and the intercept the Vmax value is 5 in micromolar into by minute, the minute cancel out and then we are having 35 into 10 to the power 5 micromolar. So this is the value of the Km. Very easy. This type of questions are very easy. Only you have to know what is the graph, how to and this graph, the derivation of my Lineweaver-Burke plot is also very easy. Okay. After getting this Lineweaver-Burke plot, just you have to put the values which are given in the question and you will get your answers. Okay. Now I will go to the next problem. Problem number 19 says, consider the plot of the concentration S divided by V0 versus concentration of S plot in the Hans-Wolf. Hans-Wolf plot is a different plot which is very similar to the Lineweaver-Burke plot. The results obtained from the graph is the intercept value is 0.25 and the slope is 0.0005 millimolar inverse. What is the Km value for this reaction? Your options are number A 5000 millimolar, number B 500 millimolar, number C 2500 millimolar and number D 250 millimolar. Consider the plot of concentration S by V0 versus concentration of S plot of the Hans-Wolf. The results obtained from the graph is the intercept value is 0.25, the slope is 0.0005 millimolar inverse. What is the Km value for the reaction? Number A 5000 millimolar, number B 500 millimolar, number C 2500 millimolar, number D 250 millimolar. I will give you a minute. Okay, since we don't have much time, let me check the chat box and then I will show you how to solve it. Okay, I have one answer which says it's number C. Let me check if number C is the, okay, I think it is for the previous question. Okay, let me show you how to solve this type of question. So the correct answer will be number B, 500 millimolar. So this is the Hans-Wolff plot. There is nothing. So this is the Michaelis-Menten plot. Just rearranging this plot will give you this equation here: y equal to mx plus c, which gives us that the c is the intercept is km by Vmax, and here the slope is 1 by Vmax. Okay. So here we have got the intercept is km by Vmax, and intercept value is given as 0.25. Okay, and the slope is given as 1 by Vmax. So 1 by Vmax is given as this one. So Vmax will be 2000 millimolar if we take the reciprocal of this one, and then Km will be your slope into Vmax. Okay, so the slope is given slope in the, so which is Vmax equal to Km by Vmax is given 0.25. Now Vmax is 2000 millimolar, so Km will be 0.25 into 2000 equal to 500 millimolar. Okay. So this Hans-Wolff plot and this Lineweaver-Burke plot both can be derived very easily from the Michaelis-Menten plot itself. Okay. Now last question of the session, it says in the Michaelis-Menten equation at high substrate concentration, the rate of the reaction becomes number A, second order with respect to the substrate concentration, number B, zero order with respect to the substrate concentration, number C, first order with respect to the substrate concentration, number D, zero order with respect to the enzyme concentration. This is a very easy question. I know all of you can answer it. In the Michaelis-Menten equation, at high substrate concentration, the rate of the reaction becomes number A, second order with respect to substrate concentration, number B, zero order with respect to substrate concentration, number C is the first order with respect to the substrate concentration, number D, zero order with respect to the enzyme concentration. I will give you a minute or two and I want all of you to answer this question. This is a very easy question and I know all of you can answer this question. Okay, let me check the chat box. I know all of you can answer the last question very correctly. Yeah, number B is the given answer. Yes, number B is the correct answer. So, when the enzyme substrate in Michaelis-Menten equation, so this is the plot. I have already mentioned it million times. So this is the at the high substrate concentration. In the low substrate concentration, it is the first order kinetics. In the middle, it is the mixed order, and at the last, it is the zero order kinetics. So the zero order kinetics means almost all enzymes are occupied with the substrate molecule, and now the reaction has become zero order with respect to substrate concentration. Okay, so B is the correct one. So this was the last question of our today's session. So today we have discussed about the nucleic acids, DNA, RNA. We have discussed about protein synthesis. Then we switched to the enzyme inhibition kinetics, and we have solved several mathematical questions also. Okay. So if you have any doubt, any query, you can let me know now. Also, I will share my email ID here in the chat box. So this is my email ID. You can save it, and you can ask if you have any doubt, any query via email also. I will try to answer your queries. Also, if you have any doubt, any query regarding today's class or previous any course content, you can let me know in the next class also. Today it was our week 3 live session, and today we have covered the week 3 course content. In our next class, what we will do is we will discuss about the week 4 course content. So right now week 4 is course content is live. Go through the content, go through the questions of the assignments, and try to solve the questions by your own. If you cannot do it, we will discuss the week 4 course content in the next class on the next Friday. After the class, you can, I think, you will be having a better view of the week 4 course content. Okay, so if you have any doubt, any query, please feel free to reach me through the email ID which I have shared in the chat box. Thank you all for joining. I will be closing the session for today. And where to find this kind of this class recording, I have already mentioned you. So you will find there is an announcement section here in the left panel. There you will find a problem solving session recording. If you click here, you will find a sheet which is basically like this. There you will find this. My name is Mousumi Deb, and you can find the course recording and the folder link here. Okay. From there, you can download the PPT which I am sharing today, and also you will find the class recording of today's. Okay. So thank you all for joining. I will close the session for today. If you have any doubt, any query, feel free to reach me through the email ID. Okay. So let's meet on the next Friday from 4 p.m. to 6 p.m. Use the same Google ID. You can save it, and you can for joining the Friday classes. Okay. So thank you all. I will close the session by today for today.